we're going to discuss introduction. Introduction to CRISPR. Like first one is an overview of CRISPR-Cas9 technology, historical background and development, mechanism of CRISPR-Cas9 genome editing, applications in biotechnology and medicines. This is which will cover unit number 1 and why genome editing. To understand the function of a precise gene or the function of the protein, okay, of the process to study its effects on the precise natures. In some organisms, it is difficult or impossible to perform site-specific mutagenesis and thereof more indirect. Indirect methods must be used such as gene silencing through siRNA complex. But in some time, gene disruption by siRNA can be variable or incomplete in process. Nuclease enzymes such as CRISPR can cut any target position in the genome to introduce a modification of the endogenous sequences for gene that are impossible to specifically target using conventional RNAi technology. Coming towards the genome editing or genome engineering are the same process, but the application also same. Using here we need to cut the DNA, cut the genome. There will be the overexpress the precise genome. Like what are the major advantages means use more recent knowledge and technology. It enables a specific area of the DNA to be modified, increasing the precision of the correction or the insertion, preventing any cell toxicity and offering perfect reproducibility. Yeah. Coming to the introduction, like how many immune system present in our body? In our body, what are the immune systems which we have?
Active and
So
passive
acquired
immunity.
immunity.
Anyone, one candidate is please. Innate immunity and adaptive immunity. Yes, correctly. What is the meaning of innate immunity? Anyone?
Natural
The
immunity.
immunity that we have by birth.
By birth. Very good. What are the barriers we have in from the birth itself?
The skin, lymph nodes, and
Then epithelium. Very good. Sweat glands, mucous membrane, followed by salivary glands and saliva. These are the various primary, various innate immune system in our body itself. And what is the adaptive immunity? Adaptive immune mechanism.
the immunity that we get through the first exposure of the antigen.
Then yes, if the exposure of antigen in our body itself, they can produce some kind of antibodies. That antibodies will interact the antigen at the time of infection, during the infection. Okay, then followed by the vaccination process. This is also best examples of adaptive immunity. Similarly, CRISPR-Cas9 is also one of the adaptive immune mechanisms in bacteria. You can consider your body is an E. coli cell. Okay, during the infection time, at the time of infection, the viral particle will infect the respective bacterium. At the time, respective host mechanism of the bacteria itself, they can produce some kind of enzymes or the proteins that particular products or enzymes or any kind of proteins, that precise protein or the enzymes where goes and interact with the viral DNA, at the time the viral particles have been break down, will be this is a normal process during the infection. Okay, like this is normal process. Similarly, at the time, this is which can combine together in the CRISPR process. Like during the CRISPR kind of infection, the bacterium itself, they can synthesize some kind of CRISPR RNA and then tracer RNA, followed by these two RNAs going to transcribe pre-CRISPR RNA, pre-tracer RNA, then these two RNA complex RNAs and followed by the RNase 3 and um, nuclease enzymes can bind together to make a complex, and that particular precise complex will interact with the viral DNA or viral RNA molecule, then the viral infection get off. This is a normal CRISPR process in the outside mechanism.